Hello, my name is Mike Shepard. I want to thank the organizers for the opportunity to present our work here at this EORTC-AACR meeting. The poster is coming from Anosi Therapeutics and presented by myself, Mike Shepard, and Dr. Pei Jin. These are the 4 important points that are made in the poster. I'll let you review those and then we'll go on to the next slide. Figure 1 compares images of a rheumatoid joint and a solid tumor. The blue arrow for the rheumatoid joint points to a structure called the pannus, which is made up of hyperproliferative and non-contact inhibited fibroblasts, sometimes called synoviocytes. It's vascularized as a result of neoangiogenesis, and it's embedded with inflammatory cells. Analogous to synoviocytes are cancer-activated fibroblasts in the tumor stroma, tumor cells, neoangiogenesis, also with embedded inflammatory cells. On the right side, I've listed some of the soluble mediators that both a rheumatoid joint and a solid tumor share. Of course, these include the inflammatory cytokines TNF, interleukin-1, and interleukin-6. But also, her family ligands have been shown to have a role in the progression of both diseases. Figure 2 shows the assembly of the ligand trap. One component is the EGF receptor extracellular domain fused to FC. The other component is the HER3 extracellular domain fused to FC. These are co-transfected, co-expressed in CHO cells, which results in the 2 homodimers, the EGF receptor homodimer shown at the top, and the HER3 homodimer showed at the bottom, and the heterodimer, which is shown in the middle. Table 1 is showing the changes in ligand binding or competitiveness for the EGF receptor, HbEGF, TGF-alpha, and neuregulin beta, neuregulin-1 beta, as a result of affinity maturation. The figure shows that we are able to obtain improvements up to 30-fold when we compare the EN220 with the EN2242 heterodimer. Panel B shows that the growth factor trap can bind either human or mouse ligands, usually with comparable affinity. Finally, in assays for ligand-induced receptor phosphorylation, we show that the increased affinity that we see in the heterodimer, the optimized heterodimer, also has improved ability to inhibit receptor autophosphorylation in vitro, in cell culture. Figure 3 shows the results from an experiment with collagen-induced arthritis mice. Inflammation in the mice was imaged using anti-E-selectin antibody with a fluorescent tag. The yellow, bright yellow color is represents high inflammation. The blue color represents low inflammation. You can see that animals treated with PBS have uniformly high inflammation in their feet. Mice treated with EN2200, the non-optimized ligand trap, have reduced inflammation. Similarly, mice treated with etanercept have reduced inflammation. When EN2200 is combined with etanercept, we see complete resolution of disease in some mice. This is also shown in the middle panel as clinical score, which is measured by redness of the joints, mobility of the mice, stiffness of the joints. The gray bar is representing the PBS treated mice. The black bar, the mice treated with etanercept, where there's some improvement. The red bar, mice treated with EN2200 with some improvement. Remarkable result is the combination of etanercept with EN2200, where the mice appear to show, some of the mice appear to show complete resolution of disease. The right panel shows the suppression of angiogenesis when CIA mice are treated with EN2200 as a single agent. There's about a 50% reduction in new blood vessels as measured by staining with anti-CD31 antibody. In figure 4, we're looking at both in vitro and in vivo studies with the ligand traps. On the top left, we're looking at inhibition of xenograft growth in a nude mouse from a lung carcinoma called H1437. It shows that in this model, the optimized ligand trap has significantly more activity than the parent native sequence ligand trap. On the right-hand side, we explore in vitro studies the ability of EN2200, the non-optimized trap, for its ability to synergize with tyrosine kinase inhibitors. On the left side, an anti-HER2 tyrosine kinase inhibitor called AG825, and on the right, an anti-EGF receptor tyrosine kinase inhibitor, gefitinib. In these models, the tyrosine kinase inhibitors synergize with the growth factor trap. On the lower left is an in vitro experiment where we compare the activities of 4 different anti-HER antibodies with the activity of the optimized ligand trap EN2242 for the inhibition of H441 tumor cell proliferation in vitro. And it's pretty clear from this result that while the anti-HER antibodies have little effect, the EN2242, which traps multiple growth factors, has a significant effect at inhibiting the proliferation of these tumor cells. The bottom right is another xenograft model called H292, another non-small cell lung cancer, where we look at whether or not a combination of EN2642, which is the oligomeric a mixture of ligand traps the homodimers and the heterodimer together, and we asked whether or not these proteins can synergize with carboplatin to inhibit the growth of lung cancer cells. The answer is yes, they do appear to synergize for this activity. This is the last slide. It's an image of our poster. Notice the QR code in the upper right-hand corner. If you image or scan the QR code, it'll take you to a link where the poster is much easier to review. Also notice that the important references for this work are also embedded in QR codes under references in the bottom right. If you have any questions, please let us know. We'd be happy to discuss with you. And thanks very much.